Welcome to today's Explainer. We're jumping right into a journey inside the microscopic machinery that literally keeps us alive, heals our wounds, and honestly, fundamentally makes us who we are. We're going to unlock the hidden world of the cell, exploring the sheer brilliance of how our cells grow, how they divide, and how they manage to generate the astounding variety of life we see all around us. So here's our roadmap for today. First up, the blueprint of life. Second, why we need new cells. Third, the cell cycle. Fourth, mitosis, making clones. And finally, meiosis, creating variation. Let's get right to it. Section 1, the blueprint of life, right inside the nucleus. To truly grasp cell division, we've got to zoom directly into the command center of a typical human cell. And I want you to just consider the sheer spatial math involved here. Inside almost every single microscopic cell in your body, there's a nucleus that's only about 6 micrometers in diameter. Tiny, right? Yet, if you were to take the DNA out of just one of those cells and stretch it end to end, it would measure roughly 2 meters long. Fitting 2 whole meters of genetic material into a 6 micrometer space, that is a mind-boggling biological packaging problem. But the body solves this packaging problem in just a totally brilliant way. It doesn't just, shove the DNA in there randomly. The DNA strands actually wind around a core of 8 special proteins called histones. Then it loops, it coils, and then it supercoils over and over again. If you remember what an old twisted telephone cord looks like, it's exactly like that. This supercoiling condenses the massive DNA molecule so perfectly that it fits inside the nucleus without tangling up into a huge mess. Just to make sure we've got our terminology straight, let's define what we're actually dealing with when the cell isn't actively dividing. We call this material chromatin. So chromatin is simply that highly coiled thread-like complex made up of about 40% DNA and 60% histone proteins. Later on, when the cell gets ready to actually divide, this chromatin is going to condense even further to form those distinct thick structures we all know as chromosomes. But what is that thread actually made of? Well, if we unravel it to its very core, we find the famous double helix. Notice how the internal nitrogenous bases pair up perfectly to form the rungs of this twisted ladder. Adenine always pairs with thymine using 2 hydrogen bonds, and cytosine always pairs with guanine using 3, always. This strict, reliable pairing is literally the foundational secret to how life manages to copy its own blueprint. Moving on to Section 2, why we need new cells, looking at the macroscopic stakes. Okay, so the blueprint is safely packed away, but why do we even need to copy it in the first place? Well, there are 4 non-negotiable reasons your body is constantly churning out new cells. First, for growth. We all started as a single cell and had to divide trillions of times to become adult humans. Second, just for the replacement of normal, everyday wear and tear. Third, to repair accidental injuries, like bridging the gap of a cut on your skin or a broken bone. And fourth, for reproduction, to pass life on to the next generation. Just to give you a sense of the sheer non-stop scale happening inside of you right at this very moment, every single second, your body destroys and replaces 2 million red blood cells. 2 million every single second. Your skin cells, they're fully replaced every 2 weeks. Your gut lining, every 5 days. You are a continuously rebuilding machine, and the whole shebang is powered by cellular division. Section 3, the cell cycle. Divide, grow, and redivide. Now that we know why this happens, let's take a look at how. Cells don't just randomly decide to split one day. They follow a highly organized, predictable series of events known as the cell cycle. And we really need to shatter a major myth right now. The idea that when a cell isn't dividing, it's just resting, completely false. The vast majority of a cell's life is actually spent in what we call interphase. During interphase, the cell is incredibly active. It's doing its daily job, it's growing, and most critically, it's preparing for the next division. Let's quickly unpack those interphase steps. In the first growth phase, or G1, the cell synthesizes RNA and proteins. Then comes the absolutely crucial synthesis phase, or S phase. This is where the DNA strand physically unzips and replicates itself completely, guaranteeing that the upcoming daughter cells will each get the exact copy of the blueprint they need. Finally, in G2, the cell makes its final structural preparations before the actual split.
Which brings us to Section 4, mitosis, making clones.
So, all that intense preparation during interphase was leading up to this, the main event, the actual mechanical splitting of the cell. The big takeaway for mitosis is understanding its core objective. Mitosis is the type of cell division where one parent cell divides into two completely identical daughter cells. The absolute most important aspect here is that the exact same chromosome number is maintained. So if a human cell with 46 chromosomes goes through mitosis, it creates two daughter cells, each with exactly 46 chromosomes. It's essentially cellular cloning, making sure your body's tissues stay stable and function perfectly. Now, the division of the nucleus itself happens in a smooth, continuous flow, but we usually break it down into 4 stages. A great trick is to just remember the mnemonic PMAT. Prophase, where the chromosomes condense and become visible. Metaphase, where they line up perfectly right along the equator of the cell. Anaphase, where the sister chromatids are rapidly pulled apart to opposite poles. And telophase, where 2 new nuclear membranes finally form around the divided DNA. There's a really fascinating structural difference here between plant and animal cells too. As an animal cell divides, it forms these star-like structures called asters to help pull the chromosomes, and eventually the whole cell pinches in the middle, creating a cleavage furrow to split. But plant cells, they have a rigid cell wall, so they can't pinch. Instead, they form a cell plate right down the middle that grows outward to separate the 2 new cells. It's the exact same genetic result, just slightly different physical mechanics. Finally, Section 5. Meiosis, creating variation. Okay, so we've seen how the body clones its cells for growth and repair. But what about reproduction? What about creating a totally new generation? This is where we have to pivot and draw a really sharp contrast. While mitosis produces 2 identical diploid cells for bodily growth, meiosis is a special kind of reduction division. It's used exclusively in the reproductive organs to produce sex cells, sperm and eggs. Meiosis goes through 2 rounds of division to produce 4 unique haploid cells. Haploid just means they only have half the normal number of chromosomes. This way, when a sperm and an egg combine, the resulting baby gets the correct full set of chromosomes. Brilliant, right? But here is, without a doubt, the most magical part of meiosis. During that very first phase, paired chromosomes from your mother and your father line up extremely closely together. And then they do something incredible. Their non-sister chromatids physically overlap, and they actually snap and swap pieces of their genetic code. We call this crossing over. It literally mixes maternal and paternal genes to create completely new, never-before-seen combinations of DNA. Which naturally leads us to a final, profound question to wrap up this explainer. If you and your siblings inherit the exact same gene pools from the exact same parents, why do you look so remarkably different? Well, the answer lies right there in the microscopic machinery we just explored. Genetic recombination and crossing over during meiosis mean that the genetic deck of cards is shuffled differently every single time. It is the microscopic engine driving human uniqueness. You are truly a one-of-a-kind combination, and now you know the exact cellular mechanics that made it happen. Keep wondering, keep learning, and we'll see you in the next explainer.